Hello, I'm Jose Antonio Sanchez Alcázar from Pablo de Olavide University. Today I'm going to talk to you about neurodegeneration with brain iron accumulation disorders, a group of ultra-rare genetic diseases characterized by iron overload in certain areas of the brain. Iron is an essential element for virtually all living organisms, but its reactivity also makes it potentially harmful. Iron accumulates with aging, and iron overload is associated with many age-related diseases in humans, Alzheimer's, Parkinson's disease. It also shortens the lifespan of several model organisms. Many life extension interventions, such as rapamycin, calorie restriction, and whole plasma dilution, can be explained by the effects they have on iron absorption, excretion, and metabolism. Iron accumulation inside the cell can be very toxic for the cells, mainly because it can form free radicals by phaeton reactions and can damage cellular components. Brain iron accumulation is also present in several genetic neurodegenerative diseases, such as Friedreich's ataxia, Huntington disease, amyotrophic lateral sclerosis, or neurodegeneration with brain iron accumulation, or NBIA disorders. Neurodegeneration with brain iron accumulation is a group of inherited neurological disorders in which iron accumulates in the brain. Main NBIA symptoms are progressive dystonia, spasticity, Parkinsonism, neuropsychiatric abnormalities, and optic atrophy or retinal degeneration. The diagnosis is usually first suspected when brain MRI findings suggest abnormal brain iron accumulation. Pathophysiologically, many of the NBIA syndromes map into related biochemical pathways and gene networks, including iron and lipid metabolism. At least 15 genes are associated with different types of NBIA. Today, we will focus on PANK2 mutations, which causes the deficiency of the enzyme Pantothenate kinase 2 activity that participates in coenzyme biosynthesis. The clinical entity is named PKAN for Pantothenate kinase associated neurodegeneration. As a first consideration, I would like to remark that not all PANK2 mutations are equal. Basically, we can distinguish between mutation with residual expression levels and mutation encoding truncated proteins. Given this presence of different mutations in PKAN patients, we started the Braincure for PKAN project with the aim of 1. To characterize pathological alteration, and 2. Evaluate the effectiveness of a library of FDA-approved pharmacological compounds. The project relies on the assumption that different mutations can contribute significantly to both susceptibility to diseases and response to drugs. The goal of this personalized medicine approach is to maximize the therapeutic efficacy and to minimize the risk of drug toxicity. As biological model, we use patient-derived fibroblasts from PKAN patients. First, we examine the main pathological alteration in mutant fibroblasts. First, we observed that PKAN fibroblasts showed a senescent morphology. PKAN fibroblasts also presented iron accumulation by Prussian blue staining. This finding is very important because we can mimic iron overload in a very easy-to-use cellular model. In here, we can see clearly the iron accumulation inside the cells. As iron accumulation is frequently associated to lipofuscin accumulation, we examined the presence of lipofuscin by autofluorescence analysis. PKAN cells showed increased autofluorescence, suggesting lipofuscin accumulation. This was confirmed by electron microscopy And very interesting, lipofuscin granules seems to be originated from mitochondria, just forming these lipofuscin granules. Currently, we are proposing the hypothesis that in many neurodegenerative diseases, such as PKAN, PLAN, and BPAN, lipofuscin granules are originated in dysfunctional mitochondria due to altered iron metabolism in these organelles. Cytotoxicity of lipofuscin is mediated by several mechanisms, including ROS formation due to the incorporation of transition metals and the ability of lipofuscin to inhibit proteolytic system, such as the proteasome. Besides cell morphology and iron lipofuscin accumulation, other important aspect is the expression levels of the mutant enzyme. As you can see here, several mutations express some amount of the mutant enzyme, but others, those encoding truncated proteins, do not express PANK2 levels at all. Once we have characterized pathological alterations, iron accumulation, lipofuscin accumulation, alteration in cell morphology, alteration in the PANK2 expression and stability, we perform drug screening in the mutant cells with different compounds that we try always that are commercial ones. The aim of this screening is to find out which drug and which drug concentration are effective in correcting the pathological alteration. In this screening, we found that supplementation with pantothenate, the substrate of the PANK2 enzyme, was able to eliminate iron in particular mutation. As you can see here, this is the iron in the patient cells, and after the supplementation with pantothenate, the iron is gone. Here you can see clearly the accumulation of iron inside the cells and how the supplementation with pantothenate is able to eliminate this iron. Pantothenate supplementation was also able to eliminate the autofluorescence of the mutant cells, suggesting that it's also eliminating lipofuscin granules. And by electron microscopy, we corroborate the positive effect of pantothenate supplementation, as you can see here clearly that lipofuscin granules are eliminated after the supplementation. As expected, pantothenate was also able to correct the senescence morphology in mutant cells. And more important, pantothenate supplementation increased the expression levels of PANK2 in a specific mutation, although it was not effective in mutation encoding truncated proteins. As PANK2 mutations are also associated with a deficiency of the transfer of 4-phosphopantothene to apomitochondrial ACP, we also examine the effect of pantothenate supplementation on MT-ACP levels. As expected, PKAN cells have low mitochondrial ACP expression levels, which were partially corrected by pantothenate supplementation in responsive mutation, but not in mutation encoding truncated proteins. Next, patient-derived PKAN fibroblasts and control fibroblasts were transdifferentiated into induced neurons by transducing lectin viral vectors containing proneuronal genes to evaluate the effect of pantothenate supplementation in one of the most affected cells in PKAN disease. After transdifferentiation, we confirm that induced neurons express neuronal markers such as tau and MAP2. and examined iron accumulation in mutant-induced neurons. As you can see here, mutant neurons have iron accumulation, and the supplementation with pantothenate was able to eliminate it. Furthermore, we corroborate that pantothenate supplementation increase PANK2 levels and MT-ACP expression levels in induced neurons. Following this approach, we have been performing personalized medicine in many PKAN mutations and testing the effects of several compounds. We have identified several supplements which are indeed able to increase PANK2 levels and mitochondrial ACP in a specific PANK2 mutation with residual PANK2 expression levels. As you can see here, pantothenate, pantethine, thiamine, carnitine, vitamin E, and omega-3 were all of them able to increase the expression of PANK2 and mitochondrial ACP and also correct all the pathological alteration in the cells. Interestingly, these supplements are also positive in patient-derived fibroblasts harboring the prevalent mutation in Dominican Republic. Recently, our group has also demonstrated in both fibroblasts and induced neurons that alpha-lipoic acid is also beneficial and improves both PANK2 and mitochondrial ACP levels in PANK2 mutation with residual expression levels. One important question is to decide between monotherapy or polytherapy in PKAN patients. Given that the selected supplements are positive in PKAN cellular models, an interesting strategy would be to evaluate the combined effect of this compound in clinical practice. Combination compounds that impact multiple targets simultaneously are better at controlling complex disease systems and less prone to drug inefficiency. The limitation of many monotherapies can be overcome by targeting disease pathomechanisms on multiple fronts. Personalized screening in patient-derived cellular models using FDA-approved pharmacological ingredients can be especially valuable because potential synergies identified by these screens can rapidly move into preclinical and clinical development. From our point of view, polytherapy or polytarget therapy has also the advantage of combining supplements with different pharmacokinetics properties. Therefore, more efficient concentration can be reached in the blood and in the brain. And what happens with PKAN patient with a stop codon mutation encoding a truncated PANK2 protein? This situation is more complicated, but we have identified several compounds which are positive in increasing MT-ACP, a key downstream protein involved in many mitochondrial functions. So we are also optimistic, and we think that also a pharmacological approach is also possible in these patients. In conclusion, from our point of view, personalized drug screening with patient fibroblasts and induced neurons can be very useful for new therapeutic approaches in patients with genetic neurodegenerative diseases such as PKAN. We think, of course, that genome sequencing, pharmacogenetics, proteomics, metabolomics, bioinformatics are very useful tools, but also we propose that response of cells to different treatments can give and provide key and essential information for the therapy of many patients. Currently, the group is looking for collaboration for conducting clinical trials in PKAN, PILAN, INAT, Cat6A, Friedreich's ataxia, and mitochondrial diseases. From our point of view, the treatment of rare genetic diseases is challenging, but is a mission possible. Several diseases are included in our Braincure platform. We are studying NBIA, disorders, ataxia, amyotrophic lateral sclerosis, Huntington disease, multiple sclerosis, etc. For getting more information about our group, you can visit our web page. Also, you can use my personal contacts, emails, and telephone to contact me if you need of my collaboration in any way. My gratitude to my collaborators which made this research possible and all the patient association who support our investigation. Many thanks for your attention.